The cells seen in outside of our body have always been the building blocks of life, from the biggest elephant into the smallest lizard. These amazing animals are made by millions to billions of cells. We, of course, are not so different from them. With absolute zero cells, we would logically not exist, because we, for a fact, were initially a one microscopic unconscious cell. But because of this essential process of the human body, we are now what we are today. I am, of course, referring to the constant and endless multiplication of cells, named as cell division. I am Shunda Peltan and I go by the name Tad. Now let's get started. First and foremost, what is cell division? Well, actually there are two types of cell division, mitosis and meiosis. Both systems are associated within a widely different task in the human body. You will soon understand how and why they occur with the screen time of my mates.
What is mitosis? Mitosis is the duplication of two or more of the same exact identical cells, the parent and the daughter cells. The contribution of this process is significant for the benefits of growth, tissue repair, and dead cell replacements. Also note that cell division does not entirely support other types of bodily cells. Some specific cells, like the skin cell, divides the day it was produced, while some cell may not divide throughout its lifetime, such as a brain cell or a neuron. Cell division, in simple words, is basically a regenerative capabilities. Just think of that time where you got cut by a blade, but then 4 days later, the cut has completely disappeared. This is the prime example of mitosis. There are multiple processes that has to occur for the completion of mitosis. This then will be explained by Mr. Vince.
The process of mitosis is a part of another system known as cell cycle. This means that there must be multiple things happening other than the process of mitosis. Cell cycle has two operating parts, interphase and M-phase, which are the mitotic and meiotic phase. Interphase is the preparatory period of cells before the M-phase occur. In this phase, cells undergo a series of different processes that ultimately leads to mitosis. Inside the cycle, there is the G1, S, and G2 phase. G0 is also present outside here. The phase, however, is under maintenance, which means that the cells in this area are not included in the cycling process. Step by step, we will get to understand these phases. Firstly, we begin at the cell growth, or as abbreviated G1. In this phase, new organelles are produced, proteins increase, and the centrioles begin to replicate, eventually create two pairs. Increase of such materials are always crucial to the cycle of cells, necessarily anyways. I think I'm already tired, Miss Christine. Can Miss Rian explain what is the next step?
Of course! Secondly, we move forward to the S phase. This part of the cycle is actually one of the most important tasks of the body, as it replicates our genetic information DNA. For basic understanding of the structures of a DNA structure are two strands of sugar phosphate backbones. These strands are also home to complementary chemical base pairs represented as A, C, G, and T. DNA also takes the shape of a double helix, or a shape that forms a sideways ladder. Furthermore, a DNA inside a cell are entirely not alone, as they are compressed together with something called octamer, which is made up of histone proteins. This DNA plus proteins is known as chromatin. DNA is extremely important as it is the body's information of how to grow, survive, and live, as well as dictates our physical features. That is why if a DNA is damaged or abnormally altered, may show some heavy consequences in and outside the body. The replication of our DNA is done thanks to the work of an enzyme called helicase. This enzyme separates the two strands, which then forms a formation called replication fork. A single DNA strand is capable of producing a new pair strands with the process of primase enzyme, in which will attach to the single strands, inviting RNA bases named primer. The purpose of these RNA bases is to act as a foundation for the upcoming construction of the new strands of DNA. The replication of one prime length is complete with DNA polymerase, binding to the primer and adding DNA bases on one direction. Specifically from the 3' onto the 5', this process continues on until the new DNA is complete.
Now, we will move on to the G2 phase. This phase is the continuation of the G1 phase. During G2, the cell continues to grow for the preparation of cell division, and the centrioles have also completed their replication and are now ready to perform the process of mitosis. After this, cell division of mitosis will begin with the divided 4 parts of prophase, metaphase, anaphase, and telophase, or for short, PMAT. The 4 parts of mitosis can also be categorized as karyokinesis. But before that, the chromatin prior to mitosis are organized and further compressed to create an equal amount of chromatin. That results to chromosomes. These organized chromosomes will then help make the process of mitosis faster. But before that, the chromatin prior to mitosis are organized and further compressed to create an equal amount of chromatin. That results to chromosomes. These organized chromosomes will then help make the process of mitosis faster. A chromosome's full structure are called chromatids, and with 2 pairs, it's a sister. These chromatids are attached to something called centromere. Speaking of which, do you know that we humans have 46 chromosomes? This exact amount can be found exactly inside the nuclei of our cells. Now that assigned in the early prophase, the centrioles that had duplicated form asters, the two also begin to move apart from one another. This aims to arrange the centrioles to opposite poles inside the cell. The nuclear membrane of the chromatin are also beginning to disintegrate. Late prophase is when the 2 centrioles and asters are successfully moved to opposite poles with the nucleolus and the nuclear membrane completely disappearing.
Meiosis is a type of cell division in sexually reproducing organisms that reduces the number of chromosomes in gametes. The sex cells or egg and sperm in humans, body or somatic cells are diploid, containing 2 sets of chromosomes, one from each parent. To produce gametes, the sperm and eggs with half of the genetic complement of the parent cells. Meiosis is a special type of cell division of germ cells in sexually reproducing organisms used to produce the gametes, such as sperm or egg cells. It involves 2 rounds of division that ultimately results in 4 cells, with only 1 copy of each chromosome.